This is a replacement for day 27, November 11, in which the camera was not recording properly from lab. And I'll take you through what we did, probably a little faster because a bunch of this you can do on your own. First comment I made was, or the discussion we had was whether to change the length of the exam. The conclusion was that I would maybe decrease it by a multiple choice and maybe a free response question. In other words, make the free response a little bit shorter. Or I would think about how can I make some of the questions a bit simpler so that they don't take quite as long. So I'm not going to make it longer. I might try to make it a bit shorter. Second point is the exam will be in ISC 1221. So just as last time, the exam will be in our lecture room on hard copy. And that will enable me to just scan them all, and it was a lot faster to grade, so I'm hoping you'll be able to get them back earlier. And during the day I went through exoskeleton problems with muscles that are flexors or sensors, so I hadn't talked that much about exoskeletons before, and some people had questions about it. Reminder about getting enough sleep, that with 6 hours or less sleep, people typically take 50% longer to solve challenging problems. It doesn't affect you at all if it's just memory or not much, but it does affect you if you have problems that actually require you to hold complex things in your head. And then the second thing is people make mistakes and don't notice. The ones where you look at later and you go, why did I choose that? Or why did I do that? And then we went through some exam problems using old exams to check understanding. So do you understand it? Using them to practice problem solving, using them to assess your transfer skills. In other words, connecting ideas from different lectures, videos, or text, different days, and related to different topics. And then we also looked a bit at change one thing and choose an outcome problems. The first thing was exoskeleton extensors and flexors. So to remind you that we're looking sideways at an exoskeleton. And so here would be the body of the animal over at this end. And the point you need to understand is where pivot points are for a particular muscle. So this would be the joints, and here, and then which muscles would cause which things to happen. I'm going to start here with the extensors. They're the ones that seem a little bit more confusing on the surface. So here is an extensor attached here and here. Here is another one. And here is another one. Now, they don't have to be on the bottom of the limb. They actually can be on the top or the bottom. So the key is you have to understand these as lever systems. So you don't want to look at this whole thing all at once. You want to look at just a part of these. And what you want to do is pick out a particular one and look at it as a single lever system. So for all of these, as I've drawn them, we've got a system that looks like this, right? So here is the pivot point. One part of the lever system and the other part of the lever system. This is the pivot point, which means this thing can go in either direction, here and here. So if we pull down on this one here, it's got to make the other one go up, right? It's going to shift this whole element here up. Similarly, if we push up here, it's going to go down. So what does the muscle do? Well, the muscle I drew goes from here to here. And of course, all it can do is shorten, right? So if it shortens, it's going to pull this down, push this up, and move our limb like this. It is an extensor. And then if we zoomed in on these others, this one is clearly the same. Here's our lever, here's the pivot point, the other arm, here's the muscle. If we pull on this, we're going to extend that. So these muscles would make the limb, extend, they're extensors. On the other hand, the flexors, this one is the same. If we pull on this, it's going to make this, it's going to make this go up. Okay, what about a flexor? And we're going to come back to this pink part that I'm drawing right here. I'm first just going to show you a muscle that attaches here and here. Here is our muscle. And in this case, if we take it out, what we have is something that looks like this. For this muscle to shorten, it can only go that way, right? It can't go this way. It can only go this way. So our muscle then is going to be pulling like so. So if our muscle gets shorter and fatter, there it is. We're pulling the limb in that direction. It's a flexor. If I drew another one just like it down here, attached at the ends, here's the pivot point, pivot points here. If we shorten this, we're going to move the limb that way. It's a flexor. You can't just memorize that extensors look like this and flexors have this extension. Because what if I put a muscle like this? If we shorten this muscle, then this limb has to go like this. We're going to bend this whole thing down. This one is also, it's also a flexor because instead of extending, it's going to pull down. So anywhere we want, we can use this structure, or we can use just a simple hinge, so a pivot point and a lever, or a simple hinge. You have to think about what is the attachment of the muscle, where is it going to move it, in order to figure out if something is going to be a flexor or an extensor. I commented that I put questions that address this issue on the exam, maybe 1 out of 2 or 3 exams. There's no guarantee you'll see one, but you should know how to work them. Then, for a part of the class, I talked about regulating contraction by calcium using either troponin, which is troponin and tropomyosin, versus calmodulin and myosin light chain kinase. And I took the class through an exercise, and you can listen to it. It's about maybe 10 minutes into the recording. For that part, you don't need any image because there was none. I had everybody close their eyes. And I walked you through the shape and using your hands in the air if you wanted to, that I encouraged, to go through the cycle of contraction with either one, whether it was actin regulated or myosin regulated. And then I asked some questions about what would be different if we made the size of the myosin head bigger or smaller. What if we changed the affinity of actins for each other, the affinity of myosin and actin, affinity of ATP, ADP, and phosphate for myosin, and so on. And for all of this, you can listen, and you just need to find the part where I start talking about regulation of contraction with calcium, troponin, and calmodulin. And it's about a 10 or 15-minute segment, and it works you through some problems. I briefly mentioned using this not to study. It will help you very little to look at this over and over. You need to practice closing your eyes, looking away, or writing on a whiteboard or on paper or an iPad, doing sketches for different parts of this or the whole thing, because that is what is going to build your memory. Looking at this makes you more familiar with it, but it does not build your memory effectively. You don't have to have sketches, so sometimes you want to test your knowledge using just words. And so here is myosin regulating actin myosin regulated contraction using myosin light chain kinase. And I have it all in words. You could add the sketches over here, or you could think through it in words and come up with solutions. And again, you can do change one things. What if there's more or less of this? More or less of this. If it's a protein, what if we make it bigger or smaller, or change the affinity of two proteins that bind to each other? I did a bacterial ruler problem. Here, I said this is smooth muscle, and I outlined one particular cell. I will try to find it again. I think it was this one. So here is the outline of this cell, going down about here. And the question was, how long is this cell? So you have what look like, excuse me, you have what look like nuclei here. Remember, muscle cells are long and thin. And so just looking at this and saying, oh, it's a cell, it must be 10 to 20 microns, you would be way off. Because remember that eukaryotic cells, while the typical ones are around 20 microns, they vary a lot in size. And of course, muscle cells are long and thin. So these are nuclei. They're typically about 5 microns or 5,000 nanometers. But these are flattened. They're skinnier this way and longer this way. So maybe they are 3 this way and 7 this way, which means that would be about 7. You could round to 10 if you wanted. It wouldn't matter, wouldn't change it much, but let's say that's about 7. Then how long is this? You ask yourself, how many of these nuclei could I fit in this length? And in class I asked, could you fit 5? And everyone said, yes. I said, could you fit 10? And some people said, no, and other people said, yes. So I said, go with 10, or if you think it won't quite fit, go with 8. So that would give you 10 times 7, that was your measurement, or our measurement here, would give you about 70 microns. Clearly much bigger than a typical cell. If we ask for the width, it's not even 10 microns. The width, as opposed to the length, the width is probably about 6 microns, even at its biggest, maybe as little as 5. Difference between striated muscles, what you're seeing here is all the sarcomeres lined up, so each of these is one sarcomere. So these actually represent the middle of the sarcomere, and so you have sarcomere, 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 sarcomere. Notice they're multinucleate, so this is all one cell with multiple really flattened nuclei, and it goes well beyond this image. This was another image that was in the notes that you could use for change one thing problems. And then we went to exams. I started with the exam from 2019. I noted that there were some questions that are not really relevant here. They were on exam 2 for us because in 2019, exam 3 had some sensory system content and so on. So this one was exam 3, although I could ask you to do a neural circuit problem. This is another sensory system 1, sensory system 1. So I wanted to jump ahead to this one. And this one, I'm going to erase this stuff. So this one is about calcium hunger. I commented that wild field mice can have a calcium hunger similar to cravings for table salt in humans. So what if we have a neural circuit that responds to the odor of bone or shed antlers from deer by causing action potentials in a neuron Y that causes mice to move toward and gnaw on the bone? So what would you expect? And if you look at these, they're all about neuron Y. So this was a complex question that is definitely a transfer problem. And I would recommend, and I probably as a student would have needed, to sketch a bit out. So we have a neuron Y. So there's Y. And it's going to trigger a neural circuit. But the question really isn't about the neural circuit. It's about neuron Y. So let's zoom in a bit on neuron Y. And what we want to figure out is, okay, for this circuit, it's got to be calcium hunger. And of course, when would you expect calcium? It would be when there's low calcium in the blood. Well, that suggests this might be connected to calcium regulation. So I might quickly jot a little sketch here. Here's the set point. And this is change over time. When we have excess calcium, that's calcitonin secretion. And when we don't have enough, that's PTH secretion. Calcitonin means you put calcium into excretory tubules and you block absorption in the gut. Parathyroid hormone does the opposite. So if we have low calcium, that would mean we're going to have high parathyroid hormone. Now, you wouldn't necessarily think through all these at once, but looking at the answers here, as you glance through them, calcitonin is one, and down here is parathyroid hormone. So that suggests these two might be involved, and certainly you'd want to know them, even if just to exclude them as wrong answers. Okay, so let's look at these possibilities. I'll do number one in green. Calcitonin opens sodium channels in neuron Y. So calcitonin then binds to get a second messenger, or maybe it's an ionotropic receptor. But anyway, you're getting sodium in, and that would be eat antlers. So does that make sense if that is true? Well, that would mean calcitonin is when you have high calcium, but in this case we have low calcium. So we can rule out that one. Next one, thyroid hormone opens sodium channels in neuron Y. Well, thyroid hormone has nothing to do with regulating calcium, and so that would be related to whether the animal had a lot of glucose or not enough glucose, or whether it was cold. It won't be related to calcium. So we can rule out thyroid hormone. What about calcium binding proteins to open potassium channels in neuron Y? Well, that would mean fewer action potentials. Okay, does that help us? Well, if there's low calcium, then we would have fewer of these. We'd have fewer calcium channels open. So with fewer calcium channels open, there's a possibility that we'd get more action potentials. Here's another one, but this time it's chloride channels, so low calcium. So we'd have more chloride coming in. With high calcium, we'd open chloride channels. With low, we'd have fewer. So that's a possibility. Next one, parathyroid hormone opens sodium channels in neuron Y. Well, parathyroid hormone happens when you have low calcium, and that one would definitely work. So best choice, parathyroid hormone, should be present when calcium is low, would open sodium channels, more action potentials, we'd trigger that neural circuit. Okay, this is a transfer problem. You have to understand this is related to calcium regulated. Cravings for table salt links back to at least some stuff related to sensory systems. Action potentials in neuron Y is back when we talked about action potentials, and some of these look at what different channel types do to change membrane potential. So that's back to membrane potential and action potentials. This answer, calcitonin, has to do with calcium regulation, hormonal regulation of calcium. Thyroid hormone is hormonal regulation of metabolism, and you need to know that's not related. Calcium binding proteins we've talked about on multiple days, and that could be involved. But remember, these calcium binding proteins would only tell you local calcium. They won't necessarily tell you whole body calcium like calcitonin and parathyroid hormone does. So parathyroid hormone, that again is hormonal regulation. So we've got material from at least 5 different days that you have to combine to answer this question. It's a transfer problem, and concept mapping can help you identify those connections while you're studying. So you see connections among multiple different days, you see the ways they are or are not connected, and then when you see a question like this, it's not so surprising to try and figure out how are all these different things connected. I think this sketch was associated with material up there. I talked briefly about a couple more transfer questions for which concept mapping can help. These don't have quite as many different days, but these are at least 3 days worth of material here. So they're also transfer problems. I briefly talked about this one to figure out how big are these. So this is part of an LH or FSH secreting cell. These are LH in vesicles ready to be released. So you can see this gray stuff. So the protein here is very tightly packed. The LH is very tightly packed, which is why it's black in an electron micrograph, but here's the membrane around it. Here is the plasma membrane. Here I labeled very thin part of a capillary epithelial cell, so we'll talk about those later. They're very thin epithelium. And here's basement membrane in between, this gray stuff, this mesh. And what I asked was, how big are these things? Well, here you have something that's about 7 to 8 nanometers. You can round it to about 10. You can ask yourself, all right, how many of these lines could I fit in here? Could I fit 10? Easily, I think. For these, easily 10. How about 20? Maybe 20. So I'm going to write down here maybe 15-ish and see what matches. Oh, so 15 times 10 nanometers, about 150. Which one of these comes closest? And down here, 100 to 200 nanometers, that comes closest. This one is well below. Micrometers is off. 1,000 to 2,000 picometers is 1 to 2 nanometers, so that is off. 5 to 10, too small. Next, I went to a few questions on the exam from 2020 and worked through these as change one thing problems. Here is the question, and you can hear on the recording, this is toward the end of class, probably about the last 10 minutes or so. I went through this question and this question and I think this question number 7. So you can hear me on the class recording while you look at this part if you want to hear the walkthrough.